So here to identify the glucose in a given sample. So the presence of sugar or glucose in a given sample of A, B, C, D. So as we know, we have to write the aim, principle, procedure and result. We have to write all these. For the principle 1 mark, procedure 3 marks and results 1 mark. So we have to write the aim as it is the question. Then principle. So here the glucose is reduced the alkaline copper sulfate of Benedict reagent. It will give color the red color, that is cuprous oxide. So they may ask the question why the red color is appeared, means you have to remember the glucose in the sample will react with the Benedict reagent. In the Benedict reagent we have a copper sulfate. It will give the red color, that is cuprous oxide. You have to remember this point. Then here spirit lamp is there, dropper, test tube and reagent, that is Benedict. So here the procedure, we have to take 4 samples into 4 test tubes. Then we can add the sample 0.5 ml or 8 drops. Then we can mix with the 5 ml of Benedict reagent. Then we can allow to boil. So then we can allow to cool. So here you have to remember, if the solution is in 0.5 color, 5% of glucose, it turns into green. Then it is allowed to yellow, it has 1%. So here the color is changed into orange, it has a 1.5%. Then the solution is brick red, it has 2% of glucose presence in the sample. Based on the percentage of sample, the color will be different. So sometimes it may get green, yellow, orange and brick red. How much percentage is there in the sample, the color is different. You have to remember the percentage and color of the sample. Identification of the presence of glucose in a given sample. So in that we have 4 different type of solutions A, B, C, D, and we can take the 4 solutions into 4 test tubes. So I will show here one sample and we have to take sample into the test tube. So in any one of the beaker. And here to know the glucose presence, we have to add the Benedict reagent to the sample. So first of all, we take the sample 0.5 ml or 8 ml of sample, and then we can add 5 ml of the Benedict reagent to the sample. Then we have to boil the mixture for 3 to 5 minutes. Then after we can allow it, it turns into the brick red color. So whatever the color, we have a different type of colors based on the concentration of the sample. It is appear in green color, yellow color, orange color and brick red color. So based on the color, we have to identify the percentage of glucose. For example, it is green 0.5%, yellow 1%. If it is in orange, it is 1.5%. If it is in brick red color, it is 2%. So here, whatever the color, you have to remember the percentage of glucose also. If maybe the external will ask the percentage based on color.